Good afternoon, everyone. Today, students, yesterday I will discuss about biological classification made by Aristotle, Carlos Linnaeus, and Horace Whittaker. Aristotle classified the living organisms based on morphological characters, and Carlos Linnaeus, father of taxonomy, he made the entire living world into two kingdoms. Carlos Linnaeus classified the entire living world into two kingdoms, kingdom Plantae and kingdom Animalia. Then Horace Whittaker classified the living world into five kingdom systems: kingdom Monera, kingdom Protista, kingdom Mycota, kingdom Plantae, and kingdom Animalia. Based on cell structure, callus organization, mode of nutrition, mode of reproduction, and finally phylogenetic characters. Phylogenetic characters means evolutionary characters. Then first, kingdom Monera. We know kingdom Monera includes all prokaryotic organisms. Kingdom Monera includes all prokaryotic organisms. For example, Archaebacteria, Eubacteria, and Cyanobacteria. Archaebacteria are ancient bacteria. They can survive in extreme environmental conditions. Archaebacteria or ancient bacteria, they are prokaryotic life forms, and Archaebacteria can survive extreme environmental conditions. See, based on the habitat, that Archaebacteria are classified into four types. Halophiles, astrophiles, thermophiles, and methanogens. So there is methanogens, methane producing bacteria. So these methanogens, the Archaebacteria, are present in the gut of ruminant animals like cow and buffalo. That's why the den of cow and buffalo are used in the manufacture of biogas or production of gobar gas. These bacteria produces methane gas, very important, methanogens. So the bacteria which are responsible for the production of the methane gas. Okay. So the Archaebacterial cell wall lack murin. Murin is present in the cell wall of Archaebacteria. Murin is absent. Lack is absent in the cell wall of Archaebacteria. But Archaebacterial cell wall is made up of glycosylated proteinaceous surface layer. In cell wall of Archaebacteria, glycosylated proteinaceous surface layer is present. Okay. So pseudomurin. So what? Pseudomurin is present. Okay. Then next, Eubacteria. Second, first one Archaebacteria, second one is Eubacteria. Eubacteria are also called true bacteria. Last class I discussed. Eubacteria is also called true bacteria because they have rigid cell wall. And Eubacteria are motile if flagella is present. If flagella is present, the bacteria are motile. They can move from one place to another place. In some bacteria, the flagella is absent. So such bacteria cannot move. Flagella is present, the bacteria is moving. Otherwise, the bacteria cannot move in the absence of flagella. So then, Eubacteria contains two types, bacteria and cyanobacteria. Bacteria consists of two types, bacteria and cyanobacteria. I have discussed the first one, bacteria. Bacteria are present everywhere. Bacteria are present everywhere. Bacteria are present everywhere. Everywhere means the bacteria are present in water and soil, contaminated food, inside our body, plants and animals. See, bacteria are present everywhere, like air, water, soil, contaminated food, and some bacteria also present inside our body, and some are present in plants and animals. And bacteria have rigid cell wall. Bacteria have rigid cell wall. Bacteria have rigid cell wall, and the cell wall is made up of, in bacteria, the cell wall is made up of murein. In bacteria, the cell wall is made up of murein. It is also called peptidoglycan. Peptidoglycan. Next. Okay, the rigid cell wall. Bacteria are rigid cell wall. The cell wall is made up of murein or peptidoglycan. Next, some bacteria are autotrophic. Some bacteria are autotrophic and some are heterotrophic. So some bacteria are autotrophic and some bacteria are heterotrophic. Clear? So autotrophic, autotrophic means the organisms can synthesize their own food material. But autotrophic, auto means self. Autotropic, the organisms can synthesize their own food material. Called autotropic. Autotropic may be either photosynthetic or chemosynthetic. So what is photosynthetic? Means some bacteria can utilize the sunlight. Some bacteria absorb the sunlight and manufacture or produce their own food material. Such bacteria are called photosynthetic bacteria. Clear? Some bacteria can absorb the sunlight and produce their own food material. Such bacteria are called photosynthetic bacteria. Some bacteria are chemosynthetic in nature. Chemo means chemicals. Some bacteria can utilize some inorganic substances and manufacture their own food material. Such bacteria are called chemosynthetic bacteria. Chemosynthetic bacteria. Clear? Next, heterotrophic. Heterotrophic. Either heterotrophic forms either parasites and saprophytes. Parasites or saprophytes. Parasites, they depend. Parasites are those, they depend on the other organisms. They depend on other organisms for both food and shelter are called parasites. Parasitic bacteria are those, they cannot synthesize their own food material. They cannot synthesize their own food material. But they depend upon other animals or host. Other animals are host for both the food and shelter. They depend upon host or other animals for food and shelter. Next, saprophytes. Saprophytic bacteria are those. They depend upon dead and decaying organisms. Dead and decaying organisms. Such bacteria are called saprophytic bacteria or saprophytes. Next, bacteria are unicellular organisms. See, bacteria are unicellular. Unicellular organisms. Bacteria are unicellular organisms with simple internal structure. But lack of nucleus. Nucleus is absent in bacterial cell. Lack means absence of nucleus. Clear? Bacteria are unicellular organisms with the simple internal structure. The internal structure of bacteria is very simple and lack of nucleus. So internal structure is very simple and lack of nucleus means bacterial cell does not contain nucleus. Means nucleus is absent in bacteria. But the DNA, deoxyribonucleic acid, DNA, universal genetic material, is present in the form of pistate. DNA is present in the form of pistate and thread-like mass. Is called nucleoid. Okay. Then see, this is the DNA. The internal structure of the bacterial cell is very simple. but nucleus is absent in the bacterial cell. But the DNA is present in the twisted. See, the DNA is present in the twisted and thread-like mass called nucleolus. Next, ribosomes. Ribosomes. Next, ribosomes. See, ribosomes are spherical units. Ribosomes are spherical units. Ribosomes are spherical units are present in the bacterial cell. Present in the bacterial cell. Present in the bacterial cell. All ribosomes aggregated. All ribosomes are aggregated. All ribosomes are aggregated during protein synthesis. During protein synthesis called polysomes. Poly means many. Poly means many. Zones, ribosomes. Many ribosomes are aggregated. Many ribosomes are aggregated during protein synthesis. This aggregation of ribosomes is called polysomes. See, polysomes. Here, the ribosomes, the spherical units present in the bacterial cell, are aggregated during protein synthesis called polysomes. Clear? All ribosomes aggregated at the time of protein synthesis called polysomes. Polysomes. Next. See, the bacterial cell, surrounded by two protective coverings. Bacterial cell is surrounded by two protective coverings. Two protective coverings. So, outermost is called cell wall. Outermost covering is called cell wall, and innermost is called cell membrane. Innermost is called a cell membrane. Innermost is called a cell membrane. Clear? See, bacterial cell is, the bacterial surface or bacterial cell is surrounded by two protective coverings. Outermost is called cell wall and inner one is called cell membrane. Outermost covering is cell wall and innermost covering is called cell membrane. In some bacteria, the third outermost covering, the third protective layer, is present. Third protective layer is present. It is called capsule. In some bacteria, the third protective layer or protective covering is present. It is called a capsule. Okay. See, clear? Two protective coverings. Outermost one is called cell wall. Innermost is called cell membrane. And in some bacteria, The third protective covering is present. It is called a capsule. Next, on the surface of the bacteria, on the surface of the bacteria, surface of bacterial cell, the surface of bacterial cell, two extensions, surface of bacterial cell, two extensions are formed. Among these two extensions, one is long, and another one is sharp. Clear? See the diagram? On the surface of bacterial cell, there are two extensions. Are formed. Among these two extensions, one is long. See, one is long. It is called flagella. Longer one is called flagella. What is the significance of flagella? In bacteria, flagella is a locomotory organ. Locomotory means the bacteria can move from one place to another place by help of flagella. So this is the longer extension is called flagella. And shorter ones are called pili. Pili is responsible for attached to host. Longer ones are called flagella. Flagella responsible for locomotion. So short ones are called pili. These are helps for attach or binding to host. Binding to the host. Clear? Binding to the host. See, there are two extensions. The long extension, long ones is called flagella. Flagella helps in locomotion of bacteria. And short ones are called pili. The pili helps in the attach or bind to the host. Okay. Next. See, the bacteria can produce endospore also. The bacteria during unfavorable conditions. During unfavorable conditions, the bacteria can produce endospore. During unfavorable conditions, bacteria can produce endospore. Then, inside the bacterial cell, plasmids means pieces of DNA. Are present inside the bacterial cell. Pieces of DNA are present called plasmids. Pieces of DNA called plasmids. So what are plasmids? See, inside the bacterial cell, the pieces of DNA are present. These are called plasmids. What are plasmids? Plasmids are defined as extra chromosomal Plasmids are defined as extra chromosomal. Plasmids are defined as extra chromosomal. Self-replicating. 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 Circular. Double-stranded. Double-stranded. Circular DNA. Circular DNA. Yeah. So inside the bacterial cell, the pieces of DNA are present called plasmids. What are plasmids? Plasmids are extra chromosomal, self-replicating, double-stranded circular DNA. Okay. So next, the cell membrane. See, this is cell wall. The cell wall component in bacteria is murin, peptidoglycan. Next, the cell membrane of bacterial cell undergo infolding. See, infoldings. In bacterial cell, this is a cell membrane. See, this is cell membrane. In bacterial cell, the cell membrane undergo infoldings to form vesicular structure called mesosomes. These vesicular structures are called mesosomes. See, in bacteria, cell membrane undergo infoldings to form vesicular structure called mesosomes. Mesosomes. Clear? In bacterial cell wall. So in bacteria, cell membrane undergo infoldings to form vesicular structures. These are called mesosomes. What is the function or significance of mesosomes? What is the function or significance of mesosomes? So mesosomes involved in cell wall formation. Mesosomes are involved in cell wall formation. They are involved in cell wall formation. Which are those mesosomes are involved in cell wall formation. And mesosomes are also involved in DNA replication. They are also involved in DNA replication and distribution to daughter cells. Distribution of DNA to daughter cells. Distribution of DNA into daughter cells. Next, so mesosomes are also helps in respiration. They are also involved in respiration. Mesosomes are also involved in respiration, and they also help in secretion. Mesosomes are also helps in secretion and increases surface area of cell membrane. Increases surface area of cell membrane. These are important functions of the mesosomes. These are the important functions of mesosomes. Here, the mesosomes in bacterial cell. The cell membrane undergo inward foldings to form vesicular structure. These vesicular structures are called mesosomes. Mesosomes play a very important role in bacteria. Mesosomes are involved in cellular formation. They also take part in DNA replication and distribution of DNA to daughter cells. And they are also involved in respiration of bacteria. They also help in secretion and increases surface area of cell membrane. These are the important functions of the mesosomes. Then, bacteria. So we know that bacteria. Some bacteria are useful. Some bacteria are harmful. Some bacteria are useful. Some bacteria are useful and some bacteria are harmful. Some bacteria helps in the production of antibiotics. Some bacteria are used in the production of antibiotics. Some bacteria are fix atmospheric nitrogen. Atmospheric nitrogen in legume plants. In legume plants. In legume plants. In legume plants. Okay. So some bacteria are used in the production of antibiotics. Some bacteria fix the atmospheric nitrogen in legume plants. Okay. So then some bacteria are harmful. Some bacteria are harmful. Some bacteria are harmful. They cause diseases too. Harmful. They cause diseases to man, plants, and animals. So they cause diseases. They cause diseases to man, plants, and animals. Okay. And the bacteria causes diseases to man. Some important diseases, for example, cholera. Cholera. Then typhoid. Typhoid. And tetanus, and so on. These diseases are caused by bacteria in human beings. Cholera, typhoid, and tetanus. These are the diseases caused by bacteria to human beings or man. So, for example, citrus canker. Citrus canker is plant disease. Is a plant bacterial disease. Citrus canker is a plant bacterial disease. So, citrus canker is caused by bacteria to plant. So, these are the important diseases. It is the bacteria can cause diseases to man, plants, and animals. See, cholera, typhoid, tetanus, these are the diseases caused by bacteria to human beings. Citrus canker is a bacterial diseases to plants. Okay. So then, reproduction. Next point is reproduction. Clear the structure of the bacterial cell. Clear? So nucleus is absent in the bacterial cell. Instead of nucleus, nucleoid is present in bacterial cell. Instead of nucleus, nucleoid is present. Means the DNA is twisted and it is in the form of thread-like mass and freely floating. This is called nucleoid. And then plasmids. Plasmids are extra chromosomal, self-replicating, double-stranded circular DNA. That is small pieces of DNA. Then polysome, aggregation of ribosomes called polysome. So aggregation of ribosomes takes place at the time of protein synthesis. This aggregation of ribosomes is called polysomes. So on the surface of the bacteria, so there are two extensions are there. So some two extensions, come on, two. One is long and another one is short. Long extensions are called flagella. Flagella helps for locomotion. And filly, short extension called filly. These are helps for binding or attached to the host. So then, during unfavorable conditions, the bacteria can produce endospore. Then, the cell membrane undergo inward folding. The cell membrane undergo inward folding to form a vesicular structures called mesosomes. The mesosomes play a very important role. They are involved in the cell wall formation. They are involved in respiration. They help in DNA replication and also distribution of DNA to daughter cells. And they also help in or involved in secretion and they increases surface area of cell membrane. Okay. Then reproduction in bacteria. Reproduction. Reproduction. So reproduction. Bacteria mainly reproduced by binary fusion. Bacteria reproduce mainly by binary fusion. Bacteria reproduce mainly by binary fusion. During unfavorable conditions, bacteria reproduced endospores. During unfavorable conditions, bacteria can reproduce by endospores. Then the bacteria also reproduce by sexual method. Bacteria also reproduce by sexual method. By primitive adopting of DNA transfer from one bacteria to another bacteria. One bacteria to another bacteria is called sexual method of reproduction. See, bacteria may reproduce by binary fusion. In unfavorable conditions, bacteria reproduce by the formation of endospores. Bacteria also reproduce by sexual method by adopting primitive transfer of DNA from one bacterium to another bacteria. In sexual reproduction, the DNA of one bacteria, clear? In sexual reproduction, the DNA of one bacteria can transfer to another bacteria. It's called sexual method of reproduction. So mycoplasma. Mycoplasma are the smallest living cells. or the smallest living cells. Mycoplasma. Mycoplasma are called the smallest living cells. And mycoplasma does not have cell wall. Cell wall is absent. Cell wall is absent in mycoplasma. Cell wall is absent in mycoplasma. These are the smallest living cells, and they can survive without oxygen. They can survive without oxygen. Anaerobic condition. Anaerobic means without absence of oxygen. And they cause mycoplasma are pathogenic for both plants, animals, plants and animals. Mycoplasma are pathogenic for both plants and animals. See, mycoplasma are the smallest living cells. The cell wall is absent. They survive without oxygen, and they are pathogenic. Mycoplasma is pathogenic both plants and animals. Example for mycoplasma. Example for mycoplasma is pleuro. Mycoplasma example: pneumonia. Like organisms. Pneumonia like organisms. PPLO. Pneumonia like organisms. For the example for mycoplasma. Mycoplasma are the smallest living cells. They lack cell wall, means cell wall is absent. They can survive without oxygen, and they cause disease both plants and animals. Best example for mycoplasma: pneumonia like organisms. PPLO. Thanks.